Hello my dear students, you are welcome. Today we are going to revise paper 1 on certain important topic, biological molecules. It's part 1. Okay, let's start. Number 1, which flowchart outlining the test for non-reducing sugars is correct? A, B, C or D? I think it's a good chance to revise how can we determine the non-reducing sugar in unknown sample. Okay, firstly we acidify the sample by adding dilute hydrochloric acid, then boiling in water bath for about 2 minutes. Then we cooling the mixtures down. Okay, then neutralization of each sample or mixture by adding sodium hydrogen carbonate powder or adding drop-wise certain strong diluted alkali like sodium hydroxide or potassium hydroxide drop by drop and check reaching the end point or the neutralization point by using litmus paper or universal indicator. It's okay. This step is so important. Why? Because Benedict solution, Benedict reagent, Benedict test works best in alkaline medium. So we have to change the mixture acidity medium to neutral, then to alkaline medium by neutralization process. Now we check each unknown sample that we predict presence of non-reducing sugar in it by adding Benedict solution, then boiling in water bath. And notes any color change or or determine the time taken to reach end point. Okay, the first color change appear in each test tube. I think all these steps match with letter C. Boil with dilute hydrochloric acid. Yes. Okay. Neutralize after cooling, neutralize with sodium hydrogen carbonate powder, then adding Benedict solution and boil. So the answer is C. Number 2, which diagrams show the release of water molecule during the formation of glycosidic bond? Here 3 figure, 1, 2 and 3 consists of 2 monomers. Which 2 monomers can combine together by formation glycosidic bond between? Look at this monomer and another one. They are alpha or beta glucose unit. They are alpha glucose. Why? Because the OH that bind with carbon number 1, okay, lie below the plane of the ring like table leg. Here, that's also alpha glucose unit, where by numbering each carbon atom, we reach to this position number 4. Here, number 1, okay, number 5, because O link between carbon number 1 and carbon number 5, so we rotate or count or numbering the carbon atoms in this direction. 1, first, second, third, fourth, then fifth one, then sixth is 1. Okay, the same for the other molecule. They are able to form glycosidic bond. This bond called alpha glycosidic bond, like this figure. Okay, 1,4 glycosidic bond. So number 1 it's okay, can form glycosidic bond. What about 2? For 2, look at the OH lie above perpendicular on the plane of the ring, so this beta glucose unit. The other molecule is beta also, where OH here for carbon number 1 lie above. How come? This down. This down due to rotation 180 degrees. Okay, to allow binding and formation of glycosidic bond. Look at the numbering now. The numbering become in the other anti-clockwise counting or numbering. Here number 1, number 2, number 3, number 4. Okay, so they are able now to form what? To form this bond. This bond called beta 1,4 glycosidic bond. It's okay. The other molecule rotate 180 degrees to be able to combine and bind by condensation process with OH of the carbon number 1 on the first sugar molecule. Okay, so 2 also is right. What about 3? 3 with the same process, by numbering each carbon atom, we reach this number 1, this number 6. Okay, alpha glucose, because OH in both that bind with carbon number 1 lie below the plane of the ring. So we can form this structure, branching like in amylopectin or even in glycogen. They are both are able to form a branching by formation alpha 1,6 glycosidic bond. It's okay. So the answer is A. Okay, answer is A. Each two monomers can combine to form a glycosidic bond. It's okay. Number 3. Which molecules have structure formula that contains C double bond O? C double bond O bonds. Amino acids. Look at the amino acid. It has structure formula that contain C double bond O. Yes, here. Look as a C double bond in this position. C double bond O. Within carboxyl group or carboxylic acid group. Okay. What about, so amino acid is right. Fatty acids. Also fatty acid contain carboxylic acid group. C double bond OOH. So it's also right. What about glycerol? Please memorize glycerol structure. It's so important in our study. Glycerol is trihydroxy alcohol containing 3 OH groups, CH2OH, CHOH, CH2OH. Okay, perpendicular in this position or horizontal like in this structure. Okay, here, where is C double bond O? No, nothing. Absent from glycerol. What about proteins? Look at proteins. Proteins, sorry, any protein molecule formed from one or many polypeptide chain. Okay, say one polypeptide chain, a sequence of amino acid as primary structure of sequence of amino acid in certain correct order. It has 2 terminals. One terminal for the first or the last amino acid in this site containing C double bond OH. Where is H? H is removed as this acid is proton donor, can release proton in medium. Okay, so leave negative charge in this O. Okay, so it also contain what? Contain C double bond O in this position. C double bond O minus charge, complete negative charge. Okay, what about the other terminal? The other terminal, N terminal, where the last or the first amino acid from the other side, okay, containing what? Containing N terminal amino group. But here NH3. Why? Because in acidic medium, in normal pH of cell cytoplasm, the NH2 can receive and can combine by proton from the cytoplasm to form this structure. Okay, now all the structures 1, 2, and 4, okay, 1, 2, and 4 have what? Have C double bond O, while glycerol with no C double bond O. It's okay, so the answer 1, 2, and 4, 1, 2, and 4, so the answer is B. Okay. Number 4. Which statement about triglycerides is correct? A, B, C, or D? Firstly, triglyceride, it is the main component of oil and fat. It is formed from glycerol molecule and 3 fatty acids, like this structure. Okay, this molecule of triglyceride. Letter A, in any triglycerides, all the fatty acids are saturated, or all the fatty acids are unsaturated. Here, by chance, the 3 fatty acids are unsaturated due double bond here in this position. It's okay. What about another image showing the first fatty acid is saturated while the other lower fatty acids are unsaturated? This one mono unsaturated fatty acid, here poly unsaturated fatty acids at position at carbon number 9 and 12 and 15. There are 3 double bonds, CC double bonds. Okay, by chance also you can find the first and second amino fatty acids are saturated, no double bonds, while the third one is unsaturated where pent or kink present between C double bond C in this position. Okay, so it is not a must, it is not a rule, so A is wrong. Okay, incorrect. What about here? This another image showing triglyceride molecule. All the fatty acids are saturated, no double bond at all. Here notes also the fatty acid bring in different lengths with different number of carbon atoms, different number of E carbon atoms. Now, letter B, triglycerides, 2 triglycerides are joined together by an ester bond. The statement is wrong. Triglyceride is monomer but large molecule. It is monomer, basic unit of oil and fat, but it is large. It is called macromolecule. But all lipids are not polymerized. They are not considered polymers. Okay, so no repetition, no joining between 2 molecules of triglycerides. Okay, so the B is wrong also. What about C? Triglycerides are polar hydrophobic. How come? Polar means polarity, means water loving. Okay, it can form hydrogen bonds with water, so the statement is wrong. Okay, triglycerides are hydrophobic, where no polarity, no partially positive or partially negative charges on the whole molecule, no complete negative or positive charges, so it is totally hydrophobic molecules. Now, for last choice, glycerol is joined to each fatty acid by covalent bond. Look at this animation several times. Here is esterification, condensation reaction, dehydration, where water removed from H from glycerol and OH from fatty acid to remove one water molecule to form ester bond. Okay, ester bond. This repeated 3 times to form triglyceride. Okay, what about ester bond? Ester bond is special type of covalent bond. Okay, so the answer is D. Look at the arrow here. Change direction between condensation and hydrolysis. Hydrolysis is the reverse due to the action of certain enzyme like lipase enzyme in presence of water. Okay, this is called hydrolysis reaction process. Okay, number 5. Many flowers produce sweet solution called nectar. Bees provided with nectar use enzyme Q to change the nectar into honey. After testing a sample of nectar for the presence of reducing sugar using standard laboratory reagents, the sample was blue. After testing a sample of honey in the same way, the sample was orange. Which conclusion about the reaction catalyzed by the enzyme Q is consistent with these results? Firstly, step by step. Okay, many flowers produce sweet solution called nectar. Yes, bees provided with nectar use enzyme Q to change the nectar into honey. So firstly, the nectar is the primary source. Look, when nectar digested in stomach and intestine of bees, okay, by certain enzyme, it change into monosaccharides. Here induced fat mechanism, here hydrolysis reaction in presence of water and certain specific enzyme to form monosaccharides. This monosaccharides, as we know, all monosaccharides are reducing sugars, while sucrose found in nectar is non-reducing, so it gives blue color at first. Okay, at start, when check nectar, it gives blue color, still Benedict with the same color, no change, so non-reducing sugar present in nectar. After digestion, after change inside digestive system of bees, it change into what? Into honey. Look, it gives positive result with different color, but the color stated here was orange. Okay, orange like this one, according to the amount or concentration of sugar present, reducing sugar present, and the test. Okay, now which conclusion about the reaction catalyzed by enzyme Q is consistent with these results? Sure, the reaction is hydrolysis. So the answer C or D. The substrate was disaccharide but non-reducing. Which one? Non-reducing. Sucrose, non-reducing. While maltose is reducing. So this wrong. Sucrose is non-reducing. After treatment with enzyme Q by bees, it changed into fructose and also glucose, digested. Even the word here, fructose or glucose, the answer is C. Okay? The answer is C. Number 6. The diagram shows the structure of part of peptidoglycan molecule. Okay, peptidoglycan means sequence of amino acids joined together by peptide bond and carbohydrate part. We hear this word before when dealing with bacteria. Bacteria cell wall is made of peptidoglycan molecule or murin, not cellulose. Okay, like this structure. Now, where is carbohydrate part? In this areas, here and here, two molecules joined together. The question now, which type of 1,4 linkage and how many peptide bonds are shown in this part of the molecule? Type of 1,4 linkage, alpha 1,4 or beta 1,4? Look, as O lie above, this means the OH of carbon number 1. Carbon number 1. Okay? For this molecule, and the carbon number 4 for the second molecule, okay, lie above the plane of this carbohydrate or glucose monomers. So it is beta 1,4. So the answer C or G. Okay, here look, as we learned before, the O atom here link between carbon number 1 and the 5. So our numbering of carbon atoms anti-clockwise 1, 2, 3, 4. Okay, here for this part 1, 2, 3, 4, 5, 6. So the link now is called beta 1,4 glycosidic bond. What about the number of peptide bonds? Peptide bonds is formed when two amino acids are joined together by peptide bond. How come? NH2 from the right amino acid, okay, donate protons, I mean H plus, and carboxylic acid group of the left amino acid, okay, donates OH. We remove water molecules as this condensation reaction to form peptide bond, okay, condensation or dehydration. So how many peptide bonds are formed in this structure, in peptidoglycan, in this part of this structure? As N link with H in this part, and as C, where is C? Yes, and as C link with double bond, okay, this is peptide bond. Where you found, or C, N link with C, C carry double bond O and N carry H, okay, this is called peptide bond. So in this tail we have to count how many peptide bonds are there. Look, the first one, second one, third one, fourth one. So the total number equal 4. So the answer is D. It's okay. Number 7. A naturally occurring polysaccharide synthesized in plant is branched chain of alpha glucose, not beta. The straight part of the molecule are linked by 1,6 glycosidic bonds with only small number of branches, okay, which are linked by either alpha 1,3 glycosidic bond or an alpha 1,4 glycosidic bond, okay. Which polysaccharide has a structure most similar to that described? Amylopectin, amylose, cellulose, glycogen. Here plant, so glycogen is not our answer, okay? Why? Because glycogen is unique to animal cells. What about cellulose? Cellulose is formed from sequence of beta glucose units, beta glucose, not alpha. So cellulose is not included with us. What about amylose? Amylose is polysaccharide. It is formed inside plant, but it is unbranched. Here he mentioned a small number of branches, so amylose is wrong also. So the remaining answer is amylopectin. Amylopectin has 1,4 glycosidic bond and 1,6 at branches, with little branches than glycogen. If we compare amylopectin with glycogen, we find that glycogen is highly branched than amylopectin. So the answer is A. Now for amylose and amylopectin, mix of them form starchy grains inside seeds or grains inside plant, okay. It is plant origin, while glycogen is animal origin, okay. It is formed inside animal cells for storage, long-term storage as energy reserve, okay, inside liver and the muscle cells, for example. It is highly branched. If we compare glycogen, okay, glycogen, where here glycogen and amylopectin, it is highly branched. 1,6 alpha 1,6 glycosidic bond with many numbers, okay, with many branches. What about cellulose? Cellulose is beta. Look at beta, O above, O below, O above, O below. Orientation 180 degrees. This is beta glucose units, okay, not alpha. What about amylopectin now, with little number of branches, okay, at 1,6 here, like this one. This carbon number 6, this carbon number 1. Alpha 1,6 glycosidic linkage with main chain, straight chain, with 1,4 glycosidic bond. Alpha 1,4. Here alpha 1,4. Here alpha 1,6 glycosidic bond. So the answer is A. Number 8. The diagram shows a triglyceride. An enzyme was used to digest this triglycerides into glycerol and fatty acids. Which scatter plot correctly represent each fatty acid component of the triglyceride, A, B, C, or G? Here's the relationship between number of carbon atoms and the number of carbon-carbon double bonds. When we digest this triglycerides by effect of certain enzyme, the ester bonds are broken by adding water. Okay, so it forms 3 fatty acids and the glycerol. Look now to the first fatty acid. Here C2. Count with me, please. C3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So this fatty acid length equals 17 carbon atoms. It is formed from 17 carbon atoms. Where is the first carbon atoms? The carbon that carry double bond O. It is part of carboxylic acid group of this fatty acid, C double bond O, OH, after hydrolysis. Now, for the second, it starts with C1 and C2, then here 17, 18, 19. So the total number equal 19. The third fatty acid, also 19. Where is number 1? Here number 1, carbon number 1, carbon number 1, carbon number 1. Okay? Now we solve the problem of number of carbon atoms in each fatty acid, the x-axis in all graphs. What about number of carbon-carbon double bond? Of sure, the first fatty acid with 17 carbon atoms with no double bonds. It's saturated fatty acid. What about the second fatty acid? Unsaturated, monounsaturated fatty acid. Unsaturated fatty acid. Here also unsaturated but polyunsaturated fatty acid. Unsaturated fatty acid. Okay, so let's check each graph. Graph A. Here number of carbon atoms for one of this fatty acid equals 16. No fatty acid with 16 carbon atoms, so this is wrong. Okay, what about B? Here one fatty acid, 1 out of 3, has 17 carbon atoms, so 17 carbon atoms, but with no double bonds, so 0 double bonds, okay, because it is saturated. What about the other 2 fatty acids? Each one consists of 19 carbon atoms. Yes. One of them carry 1 double bond. Yes, this one. And the other one carry 3 double bonds. Yes, 3 double bonds. First, second, third. So the answer is B. Okay? Okay. Number 9. The diagram shows how the alternating nature of the beta-glucose monomers along a chain allows hydrogen bonds to form between consecutive monomers. Consecutive monomers of what? Of cellulose, for example. Look, please. Here, OH, there is hydrogen bond between partially positive and the partially negative O. Partially negative O, partially positive H. Here's the same, partially positive H, partially negative O, hydrogen bond. Okay, hydrogen bond in cellulose affects the tensile strength, the ability to withstand the pulling forces without breaking. Okay, like cellulose cell wall. It resists turgor pressure when water enters by osmosis inside cell cytoplasm and vacuole. Okay, due to tensile strength, due to resistance or due to presence of this hydrogen bond. What about hydrogen bond? How do the hydrogen bonds shown in the diagram help cellulose function as suitable material for cell wall? Hydrogen bonds are longer than of covalent bond, but weaker, much weaker than covalent bond. Okay? But a lot of hydrogen bond found in cellulose molecules, this added together to form highly tensile strings. But each hydrogen bond individually is weak, very weak bond. So the hydrogen bonds and add additional tensile strings along individual cellulose molecules within the cell wall. Yes, it actually adds tensile strings. Okay, hydrogen bonds create stronger cross links. No, it cross links, yes, it is bond, yes, but it is not stronger. Okay, the description stronger is wrong. It is very weak bonds, weaker than covalent. Also stronger hydrogen bonds, not stronger. It is much weaker than covalent, as we said before. Cellulose molecules within cellulose microfibril have stronger link between them. Wrong. So the answer is A. Number 10. Two solutions 1 and 2 each contained a mix of two different biological molecules. Say you are given two beakers. Okay, two beakers. One of them contains starch and sucrose, and the other contains glucose and protein, but without labels. So we cannot detect which one contains starch and which one contains glucose. Okay. The two solutions were tested with variety of reagents to identify the presence of the biological molecules in the solution. Let's check. The table shows the results recorded for various tests. Which row identifies the two solutions? How we can determine both beakers, which one contains starch and sucrose and which the other contains glucose and protein, which and which. Look please, firstly we check by adding iodine solution for presence of starch, as we know. Okay, so it gives with one, say, positive result with A and C, while for 2 it gives a positive result for B and D. But starch is mixed with sucrose. So let's check with boiling the solution that may contain starch or sure contain starch. Boil with Benedict solution. Directly boil with Benedict solution. It sure it gives what? Negative result. So sucrose, it gives negative result when boiled with Benedict solution. So the answer, look please, 1 here gives a negative. Why not choose this other one? Because here no, with 1 here in this column, no starch present. So then we not choose this letter. So the answer till now C. What about double check for the other beaker that contain glucose and the protein? Look please, here number 1. Okay, boil with Benedict solution after acid hydrolysis. After acid hydrolysis gives a positive result, where firstly it gives negative result, but after hydrolysis, adding HCl, boiling, then neutralize, then check by Benedict solution and the boil, it gives a positive. It assure that the beaker number 1 or solution number 1 contain sucrose. Okay, contain what? Sucrose. What about the other products here? The other container here, glucose and the protein. For the second beaker, the second solution, it gives a positive result with Purette test. So 2 containing what? Containing protein. Okay, containing protein. Here also containing glucose, it gives positive and the positive either directly with boiling with Benedict solution, or indirectly after acidification, it gives also positive result. This is for glucose. So the answer is C. Okay? Number 11. The diagram shows parts of 3 pairs of amino acids within a protein. The pairs are labeled X, Y, and Z. X, Y, and Z. 3 pairs of protein. Okay? 3 pairs of amino acids within a protein. Okay. Number 11. The diagram shows parts of 3 pairs of amino acids within protein. Only each pair consists of 2 amino acids within certain protein. The pairs are labeled X, Y, and Z. Here, look, OH, OH. Oh, sure, it forms what? Hydrogen bond. Why? Because OH is polar group. Here, partially negative, partially positive, partially positive, partially negative, or slightly negative. Okay? Here, hydrogen bond will be formed. So the answer for X, B, or C. What about Y? Hydrocarbon, CH3, methyl group, methyl group, methyl group, methyl group. There is hydrophobic groups where hydrophobic interaction between them takes place. Okay? Here. What about last Z? Look at Z. Attraction forces between O that carry complete negative charge as ionic group, and the NH3 as positive ionic group. Attraction forces, this ionic bond. All of them found within protein, like this polypeptide chain, hydrophobic interactions, okay, hydrogen bond, and ionic bond. This not included in our question today. All of the 4 types of bonds found within one polypeptide chain, this allows this polypeptide chain found in tertiary level of structure. Okay? Tertiary level of structure. The answer is C. Sorry, the answer is B. The answer is B. Okay? Number 12. The diagram shows part of collagen fibril made of collagen triple helixes. The collagen triple helixes are linked to each other by one type of bond. This bond is labeled as X in the diagram. Okay, collagen fibril. Okay, collagen triple helixes or helix, like this one, this triple. Okay, triple and triple joined together by a certain bond. Of sure, this bond it is called X. Okay. What is type? Look, please. Let's revise collagen. Collagen is protein, okay, structural or fibrous protein formed from sequence of amino acids. The third amino acid, every third amino acid is glycine. Okay, this primary structure of collagen. Three of this molecule share together or bind together to form triple helix. What is meant by double helix? Double helix like DNA. It is formed from nucleotides, not from amino acids. Here, formed from sequence of amino acids. How they are joined, these helixes, joined by hydrogen bond and the covalent bonds. Okay? Sequence or parts of this collagen molecules, how they are joined together. Joined in this manner, staggered in order, but joined together laterally by cross links. This cross links not made of hydrogen bond, it is entirely a covalent bond, strong covalent bond. Okay, this staggered ends gives the striated effect seen on electron micrograph. Okay, collagen fibers found in connective tissue like tendons, ligaments, fibrous, and so on. So the answer here is A. covalent bond. Number 13, the table shows some information about the polypeptides that make up hemoglobin. Let's revise hemoglobin. Hemoglobin is globular protein. It is formed from four polypeptide chains, 2 alpha, alpha chain 1, alpha chain 2, 2 beta, beta chain 1, beta chain 2, so 4 polypeptide chains. It has quaternary structure. Why? Because it is formed from more than one polypeptide chains. At each center of polypeptide chain, there is non-protein group called heme. At the center of each heme, an ion called ferrous ion, Fe+2, that combines with oxygen at lungs. Okay? This molecule is soluble, globular, and soluble in cytoplasm of red blood cell. Okay? With huge number of millions. Okay. What about this table? How many alpha globin in hemoglobin? 2 alpha. How many beta? 2 beta. So, total number of amino acid residues in polypeptide chain, 141 for one polypeptide chain. 1. But we have 2. So, we have to multiply times 2. Equal 282. What about 2 beta? Each beta contains 146. So, 2 beta equal 292. Add all the amino acids together to obtain the sum, equal 574 amino acids in the whole molecule of hemoglobin. What about position of amino acid cysteine in polypeptide chain? Cysteine found at position number 104 in each alpha globin. So how many cysteine found in one hemoglobin molecule? Equal to how many of cysteine found in beta globin? In one beta globin, 2 at position 93 and 112. How many per one molecule of hemoglobin? Equal 4. 2 times 2 equal 4. So what about the total number of cysteine found in one hemoglobin molecule? Equal 6. Now we have to read this. Scientists studied the region of beta globin polypeptide chain containing the amino acid cysteine at position 93. They found that this region faces outwards with no oxygen is attached to the heme group, deoxygenated hemoglobin. This region faces inwards when oxygen is attached to the heme group. Replacing cysteine by mutation, for example, with different amino acids reduces pore shift. Its affinity for oxygen reduces its affinity for oxygen. What can be concluded from the information about cysteine in hemoglobin? Concluded from the information given, only the information given. More than 1% of the amino acid in one hemoglobin protein are cysteine. Let's count and calculate this percentage. Now, how many cysteine found in one molecule of hemoglobin? Equal 6. What its percentage? 6 divide percentage of cysteine, 6 divide 574 times 100 equal 1.045%. So that's greater than 1%. So the answer is A. It's okay? The answer is A. Now, number 14. Which feature of glycogen distinguishes it from starch? All glycogen molecules are highly branched. All glycogen molecules are polysaccharides. All glycogen molecules contain alpha glucose. All glycogen molecules contain 1,4-glycosidic bonds. Let's check. Glycogen compared to starch, but starch is formed from two main units, amylopectin and amylose. Here, let's check. All glycogen molecules are highly branched. Yes, highly branched. But here also branching in amylopectin. but glycogen has more branches than of amylopectin. Yes, so the answer is A. Let's check B, C, and D. All glycogen molecules are polysaccharides. Starch is polysaccharides. Okay, by its components. Okay, so it is not unique. That's wrong. All glycogen molecules can contain alpha. Here, alpha 1,4 glycosidic bond plus at branches alpha 1,6 glycosidic bond. The same for amylopectin. The same for amylopectin. Apply to amylopectin. While amylose is a polysaccharide, yes. Okay, it has helical shape but without branching. All the units with alpha 1,4 glycosidic bonds joined by alpha 1,4 glycosidic bond. So this is wrong also. Okay. All glycogen molecules contain alpha glucose, also starch alpha glucose. All glycogen molecules contain 1,4, also starch contains alpha 1,4 glycosidic bond. So the answer is A. Number 15. The diagram shows a biological molecule. Which molecules would be produced if this biological molecule is hydrolyzed? This molecule is triglyceride. It is formed from 3 fatty acids and glycerol. When it is hydrolyzed by effect of certain enzyme in presence of water, what would happen? It forms fatty acids and glycerol. Look at this presentation. Look at this figure. Adding water, breaking ester bond to form glycerol on the left and 3 fatty acids on the right. Glycerol and 3 fatty acids. So only the answer is C. Fatty acids and glycerol only. Okay. Number 16. A mixture of glucose and starch solution was placed in length of dialysis whisking tubing and the tubing sealed, like in this figure. Here, whisking tube containing glucose and starch solution, tied at both ends and put inside boiling test tube containing distilled water. Okay, the tubing was then placed in boiling test tube containing distilled water. 2 samples were immediately removed from this water at 0 time. Okay, and is tested with either iodine solution and benzylic solution. This was repeated at 10-minute intervals for 30 minutes. 10, 20, 30 minutes. Okay. What about the results? The iodine solution gave an orange-brown color each time. Look. Here about whisking tube, put inside boiling tube containing water, and we took drops of distilled water at 0 time after 10 minutes and 20 minutes and 30 minutes. And we carried out 2 experiments. The first, iodine test for starch presence. Spotting tile, we put drops of iodine and add drops of this solution at different times intervals. 0, 10, 20, 30. Here it gave an orange-brown color each time. This is the original color of iodine. So it is negative, no starch exit from whisking tube. Okay? Why? Because the starch here offshore, too large to pass through pores of whisking tube. Or the whisking tube pores are too tiny, too small to allow starch to exit from it. While glucose molecules is tiny dots, very tiny, so it released to outside by diffusion. So what may be concluded from these results? The pores in whisking tubing are too small for starch molecules to pass through. Okay? Yes, right. Glucose diffuses through the whisking tubing down diffusion gradient by diffusion from high to low, from inside to outside. Water diffuses into the whisking tube. Is it a conclusion from our results? No. we carried out 2 experiments, iodine test and Benedict test. Okay, so this number 3 is not with us, not an answer. Look at reducing test done by Benedict reagent. Sorry, at first the color still blue, remains blue. This at first, at 0 time, while at time lapse 10 minutes, then 20 minutes, then 30 minutes, it change in color and become darker. Okay, this means glucose exit by diffusion to outside. So the answer 1 and 2 only. Our conclusion, according to the information given in the question, apply to 1 and 2 only. So the answer is B. Okay, answer is B. What about this figure? Look, firstly, negative result. Here, starting positivity. Green, slightly green, then yellow, then orange, then red, brick red, according to the concentration of glucose or time lapse in this experiment. Okay. Number 17. Which molecules are globular proteins? All enzymes are globular because we need enzymes act, because enzymes act in aquatic medium. They are soluble. So amylase is globular. DNA polymerase is globular. What about hemoglobin? Look at hemoglobin. Hemoglobin found in red blood cells, in this great number of molecules inside red blood cells, in aquatic medium of red blood cell cytoplasm, must be soluble. To be soluble must be globular. It means the amino acids with R groups that faces water must be hydrophilic, and the interior or the inwards amino acids in each polypeptide chains contain R groups that hydrophobic away from aquatic medium. So the globular structure is found in hemoglobin also. So 1 and 2 and 3. Okay, so the answer is A. Number 18. The diagram shows a triglyceride. What will be the products after hydrolysis of this triglyceride? Hydrolysis means adding water in presence of certain enzyme to break ester bond. Add here OH and H. Add OH for the acid part and H for glycerol part. OH and H. How many water molecules are consumed? 3. 3 water molecules per 1 molecule of triglyceride. Okay? After hydrolysis, we obtain 1 molecule of glycerol and 3 molecules of fatty acids. So, what will be products after hydrolysis? 1 molecule of glycogen? No. Glycogen? No. So, not A, not C. Maybe B, either B or D. Look, 1 molecule of glycerol, yes. 2 saturated fatty acids, let's check. The first fatty acid is saturated. The second saturated, where no double bonds, no kink, no band. Here, unsaturated. So the answer, 2 saturated, yes. 1 unsaturated, yes. So the answer is B. Okay? The answer is B, like in this animation. Hydrolysis, breaking ester bond, okay, to form glycerol and the 3 fatty acids. Then check the type of fatty acids, whether it contains C double bond or no C double bonds. Okay. Number 19. Which description lists all the components of human hemoglobin molecule? Hemoglobin molecule, as we know, consists of 4 polypeptide chains, 2 alpha, 2 beta, 2 alpha, 2 beta, with 4 heme groups. Look, 4 polypeptide chains? Yes, 4. That are all the same? No, they are different. And one heme group? No, 4 heme groups. 1, 2, 3, 4 heme groups. Wrong. Okay. What about that are not the same? Yes, right. And one heme group? Wrong. For letter C, check C. 4 polypeptides? Yes. That are all the same? No. And the 4 heme group? Yes. 4 polypeptide chains that are not all the same? Yes. And the 4 heme groups? Yes. So the answer is D. Okay. Number 20. HIV-1 protease is an enzyme produced by the HIV virus. 2 identical chains of 99. 2 identical chains. Where? Okay. 2 identical chains of 99 amino acids form the enzyme. So this enzyme is formed from 2 polypeptide chains. It has what? Quaternary structure. It is formed from more than 1 polypeptide chain. In each chain, amino acid 25, 26, and 27 in the sequence form part of the active site. Part of the active site of this enzyme. Which order of protein structure controls the shape of the active site? Of sure, all of the levels. So letter A, primary, secondary, tertiary, and quaternary. Okay, look as this different levels. The first level, just only order and sequence of amino acids. Okay, then formation of hydrogen bonds to form alpha helix or beta pleated sheets. This form secondary protein structure. Okay, what about when extra folding for 1 polypeptide chain, only one, it forms tertiary protein structure, where some amino acids lie closer by this folding and form part of the active site, like 25, 26, and 27. What about quaternary structure? When the protein is formed from more than 1 polypeptide chain, it is considered quaternary. Okay, so look at this shape, one gray and the other one dark gray or dark or black. Okay, so it is quaternary structure. So the answer is A. 21. Which statements are correct reasons for how animals cool down in hot environments using latent heat or vaporization of water? Some animals lie down and roll in wet soil. Firstly, before completion of this question, let's define and know something about latent heat of vaporization. Look, our skin, for example, this skin, where droplets of sweat on the skin, it evaporates after few minutes. But to allow sweat droplets to evaporate in air, what allows this to evaporate easily? Absorbing heat from where? From our body. So water absorbs a lot of heat when it evaporates. Water characterized by absorption, a lot of heat content when it evaporates. It needs a lot of kilojoules to evaporate, to change from liquid phase into gaseous phase, into water vapor. So water vaporization can be used for cooling down. Why? Because it absorbs a lot of heat to be converted into water vapor. Water absorbs a lot of heat when it evaporates. When an organism sweats, the vaporized water molecules carry heat away from the surface of the body. Sure. Okay? Now, look step by step. Number 1. Some animals lie down and roll in wet soil, like this animal, oryx. Okay? It lie at shade in wet soil. Okay? At sunset, it starts activated and grazing on plants in this desert. Some animals lie down and roll in wet soil. Why wet? Because water has high latent heat of vaporization. Okay, fish move into deeper water. Fish is cold-blooded, not a mammal, not like oryx. Okay, so it is not affected by moving down deeper or above or near the surface of water. No change takes place. Okay, fish still. Also, fish live in water. Water is characterized by its no fluctuations in its temperature. No fluctuations, no very large or great variation between the water at surface and water deeper. Okay, so that's not the answer. Fish move into deeper water. Nonsense statement. Okay, what about some animals lick their fur to make it wet, like cats or dogs? Okay, they allow their fur become wet. Thus water droplets from their saliva allow to cool down their body temperature by evaporation, by latent heat of vaporization. So number 1, yes. Okay, number 3, yes. While fish move into deeper water, wrong. The last step, some animals breathe quickly with wet tongue hanging out of their mouth, like this pretty dog. Look, during panting, during running, it breathes quickly with wet tongue hanging out of their tongue to cool down its body temperature. Otherwise, the brain may be damaged, okay, and may lead to death if stop hanging out of their tongue, okay, of its hanging down its tongue. 1, 3, and 4. Okay, so the answer 1, 3, and 4. So the answer is 3. Thank you. Bye bye.